Higher level biology, paper 1a. We're going through questions 11 to 30, so this is the second video. Question 11. Under certain conditions, living organisms on Earth produce and release methane. What favours the production of methane? So methane we associated with swamp conditions, waterlogging. So that's not consistent with forest fires, so I take that one out. High light intensity, intense light, not part of the process. Anaerobic looks good. Dry conditions is completely the opposite, so our choice is 11C. The diagram shows the elements present in two organic molecules, W and X. Which molecules could W and X be? So let's just take a moment to find out about W. W has phosphorus, carbon, hydrogen and oxygen, whereas X has carbon, hydrogen, oxygen and nitrogen. So just move the question down a little bit. So this doesn't occur in monosaccharides, so that doesn't work. I'm going to go down this column first. Nucleic acids is a possibility, as are phospholipids, but triglycerides are just carbon, hydrogen and oxygen, so that doesn't work. Let's come across to the right-hand column. Amino acids are a possibility. Triglycerides are not. Protein is a possibility. Fatty acids are not, and therefore horizontally we can see that option C is our selection for this question. On to question 13 and 14. Which molecules are produced during the hydrolysis of a triglyceride molecule? Well, you'll recall that triglycerides have a glycerol molecule in this location, and then they will have, what I'm trying to indicate here are fatty acid chains. So I'm looking for glycerol and fatty acids, not this one. Fatty acids and glycerol, that seems to work. Not this one, not this one. Our best fitting answer is B. What is the consequence of the ability of water to form many intermolecular hydrogen bonds? Options. Ice has a higher density than, well, it doesn't. It has a lower density, so immediately we can eliminate A. Light can pass through water, so algae can photosynthesize. Well, indeed it can, but that is not a consequence of hydrogen bonding, so we need to get rid of B. Some small invertebrates can walk on the surface of water, so this is referring to surface tension, and we have studied a number of such small invertebrates, including the pond skater, so that looks like it's going to be our answer. Let's just check the last option. Small amounts of energy are needed to change water from one state to another. No, large amounts are. This is why we describe water as having a high specific heat capacity. Now, the reason it's worth going through the options in questions, especially when you are practicing for the final exam, is that you see alternatives and you learn information as you are going through the questions that could become useful in the questions in your own examination. At 15, what is the function of cholesterol in the fluid, in the cell membrane? And the function of this is just straight recall. It's to regulate the fluidity. You may remember the positioning of cholesterol within the cell membrane with its amphipathic structure bonded up to the phosphate groups, and also, but then merging with the fatty acid chains. It alters the fluidity of the membranes. Let's move on to question 16. And the diagram shows the movement of ions across the membrane of a neuron. Quick look at that. We've got the outside here and the inside. So notice that the sodium is going through passively. ATP, this is an active process going on here. It looks like the sodium-potassium pump. Sodium's out, potassium's in, and then we seem to have passive movement of potassium out of the cell. So let's see what the question is asking us. From the diagram, what can be deduced about the movement of sodium ions? So we're only thinking about sodium, which remember was here and here. They are actively pumped out. Well, that certainly seems to be the case. And some enter by facilitated diffusion. So that also seems to be the case. So in this case, it looks like we have option A to select. A quick look at the other ones. They are actively, so this is still talking about sodium, they are actively pumped out. Well, that's still true. And some re-enter by simple diffusion. Well, simple diffusion is through the phospholipid bilayer, so that's incorrect. So we wouldn't have that one. They diffuse out of the cell along the potassium. No, they don't. No indication of that at all. There's a net movement of sodium ions into the cell. In fact, the net movement is out. And so we are going to go with our original selection, 16A. And then 17 is relating to the structure of leucine, an essential amino acid. And it wants to know which part of leucine distinguishes it from other amino acids. Now, all amino acids are distinguished on the basis of their R group. And to locate the R group, we need to find nitrogen, carbon, carbon backbone with the R group coming off the so-called alpha carbon. So this is actually an amino group. This is actually an acid group here like this. So let's look at the diagram. So nitrogen, carbon, carbon. But the problem with this diagram is it should be like this. So we can't have A. Let's take a look at B. Nitrogen, carbon, carbon. That all seems correct. And that does seem to be isolating the R group. That's my option at the moment. Coming across here, we have nitrogen. So this is not the R group, and that doesn't distinguish it. This is actually what all amino acids have in common, so I can eliminate C. And then at this point, we're just highlighting different parts of the chain. So that's not going to work. It's actually that part there, which is the same as B. So my selection is 17B. Question 18 shows the structure of insulin. We've got a polypeptide chain here. We've got a polypeptide chain here, and they're disulfide bridge linked. What can be concluded about the structure of insulin? So this is supposed to be insulin. It's composed of two polypeptide chains. Well, that certainly seems to be the case. And they're stabilized by disulfide bonds. Well, we seem to already have our answer, but let's just check for the best answer. It's a simple protein of one continuous polypeptide chain. Well, that's not the case. Above is two polypeptide chains. It's a fibrous protein. We have no information to support that assertion, so let's get rid of that. It's a molecule. Its molecule do not display quaternary structure. On the contrary, we've got more than one polypeptide chain coming together, so that's above the tertiary structure into the quaternary, so that's wrong. 18A. Question 19 has this complex diagram. You should be able to recognize some of this. Here we've got protons being pumped into the spaces by so, and here we've got same here, another proton pump and the formation of metabolic water, and then we have ATP synthase with the formation of ATP. Energy that is released by oxidation reactions in the mitochondrial matrix is carried to the cristae on the mitochondria. How is this energy carried? So we're looking for how energy is carried to bring about this proton pumping, and we'll take a look at the options for the best fit. Well, ATP is not the case. That's actually the purpose, is to synthesize it. Glucose was all the way back in glycolysis. Enlysed water is not part of this process. And the reduced NAD, this is a great answer. This will do. And so we have our answer D. And on to our final question in this particular video. We're looking at the activity of an enzyme at different temperatures. I'll just move this slightly so you can see it more clearly. And so we have temperature on the x-axis, rate of reaction on the y-axis. We have three curves. I'm sure you may recognize all of them. But we're being asked, what does the dashed line in the graph represent? Of course, the dashed line here, what we're seeing is a decrease in the rate as the temperature increases, and this relates to the stability of the enzyme, which relates to the stability of protein, which relates to the stability and shape of the active site, and so there's a loss of the shape of the active site due to destabilization. Let's see what our options are. Increasing temperature increases the substrate concentration. There's no indication of that at all. Increasing the temperature affects the active site. Well, that's the argument that we're making here, but let's just check the other options. Increasing temperature increases the rate of reaction. Increasing temperature increases the rate of reaction. Well, that's only true up to a point where here the rate of reaction of the thing actually begins to decline. Increasing temperature decreases the movement of particles. It's actually the opposite. This will increase the movement of particles. So we are indeed left with 20B as our answer. This is the end of this video. Next video takes on 21 to 30.